We are going to study about one of the basic technique we are using in the amplification of DNA, that is polymerase chain reaction. We simply call it as PCR. So what exactly PCR does? So we have, let's say, very small amount of the DNA sample, and when we have this small amount of DNA samples, probably in micrograms, so you want to convert into the more amount for your requirements, so you have to make a multiple copies of that DNA. So at that point of the time we use polymerase chain reaction. In a crime scenes, let's say you get strands of the hair, you want to multiply, get that strand of the hair and extract DNA from it, and then you multiply the copies out of it. And once you multiply the copies of it, you have enough sample of the DNA which you could use for the multiple purpose, comparing it with the suspect's DNA samples. So this is where exactly polymerase chain reaction comes handy. So looking at the factors, the first thing I want to talk about before polymerase chain reaction, is that our body simply able to do the multiple copies of DNA by itself. Let's think about a cell going for the mitosis. So before going to mitosis, every cell needs to have 2 copies of DNA. Our cell originally does have a single copy of DNA, but when it enters to the mitosis stage, it has to have 2 copies of DNA. So here what happens, our body cell starts replicating the DNA and start making the 2 copies of the DNA with the help of one single copy of the DNA. So before entering into mitosis, every cell will have the 2 copies of DNA. After mitosis, both the cell will have the same copies of the DNA, which is single copy, which is a mother cell and daughter cell will have exactly same copy of DNA. So this is what exactly we do the multiplication of DNA in the cell, which our cell does in a day-to-day basis every day. So what are the enzymes involved with it when a cell does the multiplication or replication process? The first one is the helicase. The helicase simply breaks the hydrogen bonds between double-stranded DNA. The hydrogen bonds present between the nitrogenous bases are broken by the helicase enzyme. So once the helicase unwinds or breaks the hydrogen bonds and unwinds the DNA, so here what happens, the primase, which we also call primase enzyme, which adds the ribonucleotides complementary to the DNA strands. So once this one adds the ribonucleotides complement to the DNA strands, we will have a DNA polymerase 3, kind of a main hero of the picture, so which will adds the deoxyribonucleotides and extends the DNA strands. The extension of the DNA strands happens in both the sides. So here at the end, wherever there is a nicks are there, that will be filled up by the ligase. So these are the basic enzymes present in the DNA replication. Now when we want to talk about the PCR reaction, that is polymerase chain reaction, now let's see what exactly requirements we have. First, we need a DNA template. The template is of our choice. So basically the DNA template can be from the source of the criminal site, and the DNA can be from the plants or the DNA from the animal tissues, anywhere. So we take that template DNA, and then we have primers. The primers here we use are the synthetically prepared primers, the ribonucleotide primers we use, which are complementary, able to bind to the DNA strands. The primers, let's say I know what is the sequence I want to look forward, I might have some of the primers which are complementary to it, so I make sure that complementarity primers I'll be adding to that DNA template. And nucleotides we use here are DNTPs, we use here, and which is used for the extension. The enzyme which we have it in the cell is DNA polymerase 3, but in the in vitro, that is in the laboratory condition, we use Taq polymerase enzyme. So why Taq polymerase? Heat stable, simply because it is able to work in the higher temperatures, which is not possible with the DNA polymerase 3, because DNA polymerase 3 is heat sensitive enzyme, where exactly Taq polymerase is a heat stable enzyme, and that is why we use Taq polymerase enzyme. We use a buffer is about pH 8.0 to 9.5, and at this pH our Taq polymerase is able to work efficiently. And we have magnesium chloride. Why magnesium chloride is, the magnesium acts as a cofactor for the activity of the DNA polymerase 3. At the same time, the magnesium acts as a cofactor for the activity of the Taq polymerase 3. If this magnesium is not there, our Taq polymerase activity will be inactivated. So that's the reason we use magnesium for the PCR reactions. Now, mechanism of PCR reaction follows this way. The first one is the denaturation. So in the denaturation, let's say we have taken the sample of DNA and we have incorporated with our ingredients, that is the template, the primer, the nucleotides, the Taq polymerase enzyme, buffer, and the required factors. We have incorporated in the PCR reaction equipment. So the cycle's beginning, the first process it goes is denaturation. In the denaturation process, what happens is our temperature increases by 95 degrees Celsius. At this temperature, our double-stranded DNA, which is bonded together, nitrogenous bases which are bonded together by hydrogen bonds, will start breaking the hydrogen bonds. The temperature breaks these hydrogen bonds, which makes this double-stranded DNA into single-stranded DNA. At the same time, if we look back, the same work was done by helicase in the DNA replication process. So here the temperature breaks the hydrogen bonds, and that is a denaturation process. The second one is the annealing process. The annealing process over here what happens is the primer binds to the DNA strand. So now here the temperature drops down from the 95 degrees Celsius from the denaturation to 55 to 65 degrees Celsius. The primer binds to the DNA strand. The once it binds there, what happens is it is make itself ready for the extension process. So primers will bind, both forward primer and the reverse primers will be there. They will bind from the 5' to 3' direction. The other one is a reverse primer, which is a 3' to 5', this thing. So once it is done, the Taq polymerase, the third process is the extension, where exactly Taq polymerase starts extending the strand, extend the strand which is complementary to the template strand, using the deoxyribonucleotides which is given in the reaction mixture. And this happens at the temperature of 72 degrees Celsius. So every time the mechanism PCR follows the denaturation, annealing, and extension with the various temperature, with the various factors. So overall polymerase chain reaction, if you look at the overall polymerase chain reaction, now we have the DNA template. The template has 5' direction to 3' direction, the anti-parallel 3' to 5' direction. So first one, we go for the denaturation, where exactly the temperature is around 95 degrees Celsius. The breaking of the hydrogen bonds happens by the temperature. So once the breaking of the bonds happens, the second process is annealing. The annealing process, where exactly the primers will be added. One is forward primer, the other one is the reverse primer. The forward primer will be of 3' to 5' direction. Forward primer is of 5' to 3' directions. The other one is the forward primer is of the 5' to 3' reaction, and the reverse is of the the other strand, which is of 3' to 5' direction. So then we have the extension. The extension happens by the Taq polymerase enzyme and at the temperature at 72 degrees Celsius. And this cycle, so this is a one cycle. Let's say we have one copy of the DNA, and the end of the cycle we will have 2 copies of DNA. Now same thing, now if we repeat the 2 copies of the DNA, if we double it up, it will become 4 copies of DNA. Now 4 copies again while run the same cycle, it will become 16 copies of DNA. So every time if we double it up, so every time we run one cycle, which is following up denaturation, annealing and then extension, we will have a doubled amount of the DNA copies. So this is what happens at the end of the chain reaction. So we can have how many chain reactions, how many ever reactions we can have, the cycles we can continue it. So short, the DNA polymerase chain reaction goes this way: denaturation followed by the annealing and then extension. Application of the PCR. PCR is used in the research for the amplification of DNA. Let's say the sample is extracted is in the small amount, the amplification is done by the PCR. And to study about the DNA markers, that is to understand about the primers, we use the polymerase chain reaction. Forensics. In the forensics, basically the DNA sample obtained in the place, the crime scene, used for the amplification. So at the end of the day, you have a multiple copies of it, for continuing or for carrying out the any amount of experiment using the same samples. And then medical diagnosis, we use the PCR for the testing of the viral and bacterial infection also. Usually the RT-PCR is used for the viral if the viral contains RNA as a DNA genetic material. Advantages of PCR, which is highly specific, it is rapid and sensitive at the same time. The limitation of PCR: errors, inhibition and target material. So what is it in the common sense is like the limitations of PCR, we use Taq polymerase has its mistake during extension, leading to the false result. In the inhibition, certain inhibitors might be present in your reaction mixture, which will not be allowing it to carry out the cycles, that is denaturation, annealing and the extension processes. The target material, so here what